Hi, everyone. This section of the lecture is going to focus on prokaryotic cell structure and function. So prokaryotes make up bacteria and archaea, and understanding them is so important to understand things like how we treat bacterial infections. So by the end of this section, you'll be able to identify prokaryotic cells. You'll be able to identify the components of them. You'll be able to describe the structure and function of everything in prokaryotic cells. You'll understand the components of the cell wall in bacteria and why it's important. You'll be able to differentiate gram-positive bacteria, gram-negative bacteria, Mycoplasma bacteria in terms of the cell walls, whether they're present or they're not, as well as archaea, which are not bacteria. And you'll be able to identify the different shapes of bacteria that we call morphologies. So this is very important to understand the differences between bacterial cells and human cells. Again, bacterial cells are prokaryotes, human cells are eukaryotic, and this one will focus on prokaryotic cells. Okay, so the outline for this lecture is we're going to talk about every component in a prokaryotic cell, its structure and its function. And as a reminder, bacteria and archaea are made up of prokaryotic cells. So when someone has a bacterial infection, this is what we're focusing on. And when people try to create treatments for bacterial infections, they try to look if there's anything unique in the bacteria in terms of their plasma membrane or their cytoplasm or their ribosomes or their cell wall, if bacteria even have it, a capsule, which may or may not be present, flagella, which also may or may not be present, as well as other structures like endospores. So this is just an overview, and we're going to focus on each of these structures, which play a role in bacteria's ability to survive, adapt, and cause disease. Okay, starting off with the plasma membrane, which is also called the cytoplasmic membrane and also called cell membrane. They all mean the same thing. The plasma membrane of prokaryotic cells is made up of a phospholipid bilayer, and the phospholipid bilayer has proteins integrated in it. We say it's a phospholipid bilayer because it's bi, meaning two. It's made up of two layers. There's hydrophilic heads, and these face outwards towards the external environment and towards the cytoplasm. And there's hydrophobic tails, which are inward, and this prevents most substances from freely going across the membrane. So the membrane is described as a fluid mosaic model. That means that the cell is generally fluid, and it talks about the fluidity of the lipid bilayer. And there's also proteins embedded within the plasma membrane, and these are important because it helps the membrane transport things in and out of a cell. So the function of the membrane, the reason why all cells, not just prokaryotic cells, have a plasma membrane is the plasma membrane is mainly for protection. So it acts as a physical barrier to protect all the cell's internal components, like the DNA and everything else inside the cell. So it's for protection. It's also used in transport. So it regulates the movement of things in and out of a cell through what we call selective permeability. Selective permeability means it doesn't let everything in and out. It selects what can and can go in and out. So small nonpolar molecules can pass freely, but larger or charged molecules require transport proteins. So that's why there's proteins in the cell membrane. The membrane is also used for sensing the environment. So the membrane has receptor proteins that detect changes in the environment so that cells can adapt to different conditions. And finally, for prokaryotes, like for bacteria, the plasma membrane plays an important role in producing energy. In cells, the energy currency is called ATP. It's a molecule that we'll learn about later. So in medicine, sometimes targeting the plasma membrane is important, and some antibiotics like polymyxins work by disrupting bacterial membranes. And here's a small video about the cell membrane.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and two fatty acid, which face the core of the membrane, and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain two types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
So that is a cell membrane. Again, with the cell membrane, it's there to protect the insides of a cell, and it's made up of a phospholipid bilayer, and there's proteins across the cell membrane to help things in and out of a cell. And we'll talk about transport later in its own section. Now we're going to talk about the bacterial cell wall or the prokaryotic cell wall. So we just discussed the cell membrane. All cells in the world have a cell membrane. Outside of a cell membrane, there's something called a cell wall. Cell wall and cell membrane are separate things. All cells in the world have a cell membrane, which is what we discussed. In addition to a cell membrane, some cells also have a cell wall. So the bacterial cell wall or prokaryotic cell wall is outside the cell membrane. So what it does is it provides another layer of protection for organisms that do have a cell wall. It prevents what we call osmotic lysis. We'll learn about that later. It prevents cells from basically bursting. It protects the cell membrane, and you can envision that if bacteria do have a cell wall, it's harder to treat them. So they're more pathogenic. Pathogenic means they cause more disease, and we'll learn about that word later. So the bacterial cell wall is made primarily of something called peptidoglycan. Peptidoglycan is made up of something called NAG and NAM molecules. NAG stands for N-acetylglucosamine, and NAM stands for N-acetylmuramic acid. So again, the cell wall is made up of peptidoglycan. Peptidoglycan is made up of NAG and NAM molecules, and these are cross-linked together and form this rigid structure that helps the cell have its shape and also helps cells withstand pressure. So that's what I wanted you to get from this slide is just if I ask you what's the cell wall made up of, it's made up of peptidoglycan. Peptidoglycan is made up of NAG and NAM, and there's a lot of antibiotics that actually work to target NAG and NAM molecules in the peptidoglycan to target bacteria. Keep in mind that the cell membrane, which is different than the cell wall, was made up of a phospholipid bilayer with proteins. Okay, in bacteria, we can separate most bacteria into two categories called gram-positive and gram-negative bacteria. These refer to their cell wall structure. And if anyone is wondering, where did the word gram come from? Gram comes from the word of the scientist who invented a staining procedure. And we realize that when we stain bacteria, they either look gram-positive, meaning they look purple under a microscope when you do something called a gram stain, or gram-negative where they stain red. So gram-positive and gram-negative bacteria differ primarily in their cell wall structure. Gram-negative bacteria, which are right here, have a thin peptidoglycan layer and an outer membrane, and the outer membrane makes them more resistant to certain antibiotics. Gram-positive bacteria have a thick peptidoglycan layer, and so it has a lot of layers of peptidoglycan, which helps them retain staining during a gram stain, giving them a purple appearance. So the cell wall of both is made up of peptidoglycan. The one that has thick peptidoglycan is gram-positive bacteria. Thin peptidoglycan is gram-negative, and outer membrane is gram-negative. And here are the lipopolysaccharide. So the outer membrane of gram-negative bacteria has something called lipopolysaccharides or LPS. And this is important because it makes bacteria harder to treat. And this will be another layer of pathogenicity that we'll talk about later. So the outer membrane of gram-negative bacteria contains lipid A as well, which is an endotoxin that can trigger a really strong immune response in humans. So this difference is very important clinically if you have a gram-negative infection versus a gram-positive infection. Focusing on gram-positive bacteria like Staphylococcus and Streptococcus, we can think of like staph infections or strep infections, have a thick peptidoglycan layer, which is most of their cell wall. And I want you to know that I don't want you to memorize that these are gram-positive bacteria. I'm just giving you an idea of some bacteria that you may have heard of, like a staph infection or a strep infection. These bacteria are gram-positive. It means that they have thick peptidoglycan. So this thick peptidoglycan is a target for many antibiotics, and the teichoic acids are also present in the cell wall, which help maintain structure and is important for the bacteria. So that's gram-positive bacteria. Next, we have gram-negative bacteria. Gram-negative bacteria are things like E. coli or Pseudomonas aeruginosa, which causes bad lung infections. Salmonella causes food poisoning, and many, many others. They have a more complex cell wall. Their cell wall is made up of a thin peptidoglycan, like one layer usually, and they also have an outer membrane. And the outer membrane provides an extra layer of protection, making gram-negative bacteria more resistant to antibiotics. So the thin peptidoglycan layer, and then there's an outer membrane, and then in the outer membrane contains lipopolysaccharides or LPS. So LPS, especially the lipid A portion, is an endotoxin that triggers immune responses in us, and this sometimes can lead to septic shock, which is a serious thing in hospitals. So that is gram-negative cell walls. Gram-negative cell walls are made up of thin peptidoglycan plus an outer membrane, and the LPS lipopolysaccharides make up a portion of this, and it is an endotoxin. So just keep that in mind. So we briefly talked about gram staining. So the gram stain is one of the most important techniques in microbiology. It differentiates bacteria based on their cell wall structure, whether they're gram positive, they have a lot of peptidoglycan. And I always tell students memorize PPP. So positive peptidoglycan, purple. That's how you're going to memorize it. So if I tell you what color do gram-positive bacteria stain purple, what's special about them? Lots of peptidoglycan. Positive peptidoglycan, purple. Gram-negative bacteria stain red. They have a thin peptidoglycan layer and they have an outer membrane. So that's the difference between them. Gram-positive bacteria, because of their thick peptidoglycan layer, they retain the purple dye, which is called crystal violet. And I'm not going to talk a lot about the gram stain because we do it in lab and you learn about it in lab. For gram-negative bacteria during the gram stain, the alcohol that comes, so you have crystal violet, then you have iodine, then you have alcohol. The alcohol dissolves the outer membrane and removes the crystal violet. So these bacteria are counterstained with safranin, which makes them appear red. So this is an important procedure in clinical diagnostics. Okay, now we're going to talk about something different. So we talked about gram-positive bacteria and gram-negative bacteria, and most bacteria either fall as gram-positive, they have a lot of peptidoglycan, they stain purple in lab, or gram-negative, meaning they have thin peptidoglycan and they have an outer membrane and they stain red. There's another type of bacteria called Mycoplasma bacteria, and Mycoplasma are neither gram-positive nor gram-negative. They're unique in the prokaryotic world because they do not have a cell wall. Instead of having a cell wall, they have sterols in their plasma membrane, which gives them some structural support. And without a cell wall, these bacteria are resistant to a lot of antibiotics that target the cell wall. For example, penicillin penicillin is an antibiotic that targets the cell wall. You would not prescribe it for anyone who has a Mycoplasma infection because they don't have a cell wall. So if you give an antibiotic that targets the cell wall and there's no cell wall, the bacteria are going to continue doing the infection. Mycoplasma pneumoniae is a specific type of Mycoplasma bacteria that causes something called walking pneumonia. Walking pneumonia is a milder form of pneumonia. Pneumonia is a lung infection, and this typically affects children. Because there's no cell wall, infections that are caused by Mycoplasma must be treated with antibiotics that target things like protein synthesis or anything that's not the cell wall. So would you do a gram stain with Mycoplasma bacteria? No, because they don't have a cell wall, so it would be kind of pointless. So now we did gram-positive bacteria, gram-negative bacteria, both have a cell wall. Mycoplasma bacteria, no cell wall. And an example of Mycoplasma bacteria is Mycoplasma pneumoniae, which causes walking pneumonia, more commonly in young children. Okay, now about Archaea briefly. So this whole lecture is about prokaryotes, and bacteria and Archaea are made up of prokaryotes. So Archaea, like bacteria, are prokaryotic, but they differ a little bit in their cell wall structure. Unlike bacteria, Archaea do not have peptidoglycan in their cell walls. Their cell walls have something called pseudopeptidoglycan, and they have other unique structures. So pseudo means fake. So we say that they have a pseudomurine cell wall, or they have a wallless, they're wallless organisms. So Archaea are usually microbes that are usually found in extreme environments, but they can be found in other environments. And their cell wall structure allows them to survive in these harsh conditions, like environments where there's high salt or it's very hot. So from a medical perspective, Archaea are not really known to cause human disease as of now. So understanding their structure helps us understand prokaryotic life, but we're still learning a lot more about them. But the take-home message from here is that their cell walls, if they do have them, are not made up of peptidoglycan. They're not like bacteria. Okay, a little bit about penicillin. So penicillin is an antibiotic, and penicillin is one of the earliest discovered antibiotics, and it works by stopping enzymes that help in the formation of the cell wall, of the peptidoglycan cell wall. So if you attack the peptidoglycan cell wall, the cell wall becomes weak and the bacteria eventually bursts because you end up with osmotic pressure. So that's why penicillin is very effective. And because it specifically targets peptidoglycan, it stops the cross-linking of NAG and NAM molecules in peptidoglycan. Penicillin is a lot more effective in gram-positive bacteria because they have a thick peptidoglycan. In gram-negative bacteria, they're more resistant to penicillin because they have an outer membrane, and this prevents the antibiotic from reaching the cell wall. This is why doctors need to be cautious about what antibiotic they prescribe depending on the infection. So if someone has a gram-positive infection, penicillin would work great. If you have a gram-negative bacteria, it wouldn't really make sense to prescribe something that targets peptidoglycan because, as a reminder, gram-negative bacteria also have an outer membrane. Now, penicillin, I just said, targets peptidoglycan. Can it treat walking pneumonia? Walking pneumonia is caused by Mycoplasma, and I just said Mycoplasma do not have a cell wall, meaning no peptidoglycan, so it would not treat walking pneumonia. Archaea, their cell wall is not made out of peptidoglycan, so it also wouldn't be able to treat Archaea. So just that idea with that. Okay, another new structure we're talking about is the bacterial capsule. So some bacteria may or may not have something called a capsule. So some bacteria have it, some don't. Bacterial capsule is an additional protective layer that's found outside the cell wall. So if bacteria do have a capsule, first you'll have the plasma membrane, then you'll have the cell wall, then you'll have a capsule. And this capsule really serves an important role for bacteria and protecting them. In terms of structure, the capsule is kind of like this gel-like covering that's made up of polysaccharides. Polysaccharides is a fancy word for sugars, and they contain some peptides. Peptides is a fancy word for proteins. We'll learn about that later. And capsules play a very important role in virulence of bacteria. Virulence means their ability to cause disease, similar to pathogenicity, because it allows bacteria to hide from your immune system. So, for example, there's a bacterium called Streptococcus pneumoniae, and it's a common cause of pneumonia. So, this causes a type of pneumonia that's hard to treat because the capsule prevents your own immune system from recognizing and attacking the bacteria. So, medically, bacterial capsules are so important in infection because they make infections much worse. And in terms of their function, they help bacteria attach to surfaces. So capsules allow bacteria to stick to surfaces like your teeth, tissues, and it helps bacteria establish infections. They also help bacteria hide from the immune system, so they protect bacteria. And phagocytosis, we're going to learn about this word later. This is when your immune system eats up microbes. Capsules sometimes can prevent recognition and destruction by white blood cells that do phagocytosis or eat up the microbe. Next, we have flagella. Some bacteria have flagella, some don't. Flagella give bacteria the ability to be motile. So flagella are these structures that extend from the bacterial surface and they allow for movement. Bacterial flagella rotate, and this helps bacteria move towards their environment, like they want to move towards nutrients or away from harmful things like white blood cells. So in bacteria that cause disease, motility is so important for infection. So the thing that gives bacteria motility is flagella. When we see flagella all around the cell, we call it paratrichous. When there's one on one end, we call it monotrichous. When there's a lot on one end, it's called lophotrichous. And when there's flagella on both ends, it's called amphitrichous. This right here, paratrichous, we see it commonly. E. coli, the flagella arrangement of it is paratrichous. It's all around the cell, and E. coli are motile. So anytime a cell has flagella, it's motile. Again, they're external filaments. They're made up of protein. So flagella are made up of a protein called flagellin, and they're not present in all bacteria. And if they are present, they help with bacterial motility. And here's a small video on it.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Paratrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
So that's flagella, and you can envision that it really helps bacteria with causing worse infections. So there's a lot of drugs being created to try to target bacterial flagella. Next, we have something called Fimbriae and pili. They're not really related, but I put them on one slide. So I'm going to start with explaining Fimbriae. So Fimbriae are these short hair-like structures found on the surface of many bacteria. So is pili. They may or may not be present. And Fimbriae are primarily used for attachment. So bacteria use Fimbriae to stick to surfaces like our own tissue, which is important for infection. For example, there's a bacterium called Neisseria gonorrhoeae, which causes gonorrhea, which is a sexually transmitted infection. And these bacteria use their Fimbriae to attach to someone's urogenital tract, and they cause gonorrhea. So that's what Fimbriae is for, mainly for attachment to host tissue. Pili are a little bit longer than Fimbriae, and there's not as many of them. They're involved in the main thing they're involved in is something called conjugation. Conjugation is where bacteria have the ability to exchange genes between each other. We'll learn all about it later. So that's when one bacteria can transfer a gene to another bacteria. Think of it this way. Let's say you have a bacteria and it has a gene that makes it resistant to an antibiotic. If there's another bacteria nearby and they have pili, they can do conjugation, meaning one bacteria can give a copy of its gene to the other bacteria. And this is one reason why we have so much diversity in the bacterial world, as well as why resistance can spread so quickly. So we'll talk about that later. But this is why pili and Fimbriae are important. So pili is what bacteria do to exchange genetic material. We call that conjugation. Fimbriae is used for attachment, and there are both these like hair-like structures. Next is the cytoplasm. I would say cytoplasm is just a fancy word for everything inside the cell. So the cytoplasm is where basically the inside of the cell. So you have cell membrane, and then inside of the cell is the cytoplasm. Cytoplasm is the watery part, which is called the cytosol, and it also has something called the cytoskeleton. So the cytoskeleton, okay, actually, I want to talk about this a little bit. So even though prokaryotes don't really have as complex of a cytoskeleton as eukaryotes, they still have some proteins that help maintain the cell's shape. So the cytoplasm is where all these things are happening. Now, prokaryotic cells, bacteria and archaea, do not have a nucleus, but they do have, there is something called a nucleoid. And I want to make it clear right now that nucleus and nucleoid are very different things. So prokaryotes, which is what this lecture is about, do not have a nucleus. But prokaryotes, the area where the DNA is called the nucleoid. So the nucleoid is just a fancy word for the region or the location in the cytoplasm where the bacterial DNA is located. Bacterial DNA and bacterial chromosome mean the same thing. So chromosomes are DNA, so we use them interchangeably. So unlike eukaryotes, again, bacteria, prokaryotes do not have a nucleus, but their DNA, the region where their DNA is, that's just called the nucleoid. Next, we have plasmids. So plasmids are these small circular pieces of extra DNA that some prokaryotes, some bacteria have. They usually carry genes that give an advantage to the bacteria, like antibiotic resistance or toxin production or the ability to use different nutrients. Bacteria can have 0 plasmids. They're just extra pieces of DNA. They can have 1, they can have 2, they can have many different types of plasmids. So plasmids are very important in medicine because they can be transferred between bacteria, for example, through conjugation, which is what pili do. So this is why they're important, and we will learn all about plasmids later. But right now, I just want you to know they're extra DNA. Sometimes we call that extra chromosomal DNA. Next, one of the most important things of cells is ribosomes. Ribosomes are the structures in cells that make protein, and protein is important for everything in an organism. So the function of ribosomes is to make proteins. In prokaryotes, ribosomes are made up of a 70S ribosome. The 70S ribosome is made up of a large subunit and a small subunit. The small subunit is called the 30S ribosome, and the large subunit is called the 50S ribosome. Now, you may have noticed that when I say 70S ribosome, so we call bacterial or prokaryotic ribosomes 70S ribosomes. You may have noticed that I just said that the 70S ribosome has a 30S small subunit and a large 50s subunit, and 30 and 50 do not add up to 70. That's because it has nothing to do with adding them to get the whole ribosome. S just has to do with when you do something called sedimentation. So when you basically spin these up in a centrifuge, it has to do with how fast they settle. So that's where the S comes from. So it's sedimentation rate. So all I want you to get from this slide is all prokaryotes have a ribosome. We call the whole ribosome a 70S ribosome. And in the 70S ribosome, it's made up of a 50S large subunit and a 30S small subunit. And the reason why this is important is in the future, you're going to learn about antibiotics. So there's antibiotics that target the 70S ribosome. And so these antibiotics are very effective against bacteria because bacteria have a 70S ribosome. And now we have endospores. So some bacteria may form something called endospores. Endospores are these very resistant dormant structures that are formed by some bacteria like Clostridium and Bacillus, and they make them due to stressful conditions. So endospores are these structures that can survive extreme heat or high radiation, or maybe there is no nutrients. So they allow the bacteria to remain viable for long periods. So for example, if you have a bacterium that has the ability to form endospores, and let's say the environment all of a sudden becomes very stressful, meaning that it's very hot, there's no nutrients. Instead of the bacteria dying, it will form an endospore. And these endospores are very resistant. So it's almost like a hibernating form of the bacteria. They can survive for thousands of years. And when the conditions become favorable again, like there's nutrients again, it's not extremely hot, all these stresses are gone, the endospore can germinate again and return to its active or what we call vegetative state. So endospores can survive very, very tough conditions, and they're a major concern in healthcare and hospitals because they're very difficult to destroy. So that's why in hospitals you have to autoclave and you have to use very, very strong sterile techniques. You can't just clean things with Clorox because we're worried about endospores. And this is why infections caused by bacteria that can form endospores like Clostridium difficile are very hard to get rid of. And this causes things like colitis in hospitals. So a little bit more about endospores. So sporulation and germination are the processes that allow bacteria to switch between active and dormant states. So sporulation is the process of forming an endospore. Sporulation comes from spore. So think sporulation, make a spore. This is when environments or conditions are bad, so the endospores are formed when the environmental conditions are not favorable. And during this process, the bacteria's DNA is put into a tough protective coat. And then if the conditions improve, the endospore can germinate, meaning that it can return to an active or vegetative state and resume normal cellular activities. So this is very important to understand. So that's what that is. And remember that endospores is not a form of reproduction for bacteria. And finally, we're going to talk about the morphology of bacteria. Bacterial morphology is a fancy word for shape. Bacteria are very small. They're measured in micrometers. Bacteria have distinct shapes, and these morphologies are very important in identifying bacteria, especially when we think clinical and research settings. Like someone has a sore throat, you take a sample. If you look at it under the microscope and you see a specific type of morphology or shape, you might have a better idea of what type of infection they have. The three most common shapes are coccus, bacillus, and spiral. So bacillus just means rod-shaped bacteria, and coccus means circular-shaped bacteria, and then spiral bacteria are spiral. You don't have to memorize what bacteria fall into each, but just to give you an idea, E. coli is a rod-shaped bacteria. For coccus or circular shaped bacteria, there's staphylococcus, which can cause bad skin infections. And then For spiral, there is bacteria like Vibrio cholerae, which causes the bad diarrheal disease called cholera. And then when we have things in pairs, we call them diplo. When they're in chains, they're called strepto. And when they're in clusters, we call them staphylo. So if I tell you, how can you identify Streptococcus bacteria with a microscope? Streptococcus bacteria can cause strep throat and other things. Strepto means chains. So I know if I look at it under a microscope, they're going to be in chains, and coccus means circular. So this is what Streptococcus bacteria would look like. So if I see that, I would have an idea that it's most likely Streptococcus bacteria. So here we have a picture of gram-stained Bacillus anthracis. So this image shows this bacteria. This bacteria is gram-positive, and the reason why I know it's gram-positive is because it's stained purple and it's rod-shaped, and so that's why it's important to understand how to read images. So just if someone came to me and they had a really bad lung infection, and you took a sample and you did a gram stain and you saw this, it would give you a big hint that it's Bacillus anthracis. I would know it's gram positive because it's purple, and the rod-shaped morphology gives it away. So that is the end of our prokaryotic cell lecture. So in this lecture, we covered the plasma membrane. It's important for protection. It's selectively permeable. It lets things in and out. Cytoplasm is the inside of the cell. Nucleoids is just the region where DNA is. Prokaryotes have a 7DS ribosome. Ribosomes are important for making protein. Some bacteria have a cell wall. If they do, it's made up of peptidoglycan. Gram-positive bacteria have thick peptidoglycan. Gram-negative bacteria have thin peptidoglycan and an outer membrane. Some bacteria also have a capsule. Some bacteria have Fimbriae. Fimbriae helps with attachment. Some bacteria may or may not have Flagella, which is for motility. Pili are used to exchange genetic material, therefore conjugation, and some bacteria may form endospores when environments are stressful. So by the end of this lecture, I'm hoping that you are able to understand these things. So I'm hoping that you're able to list all the components of a prokaryotic cell, describing their function. I would hope that you're able to understand the structure of the bacterial cell wall, differentiating gram-positive, gram-negative cell walls. Again, gram-positive bacteria have thick peptidoglycan. They stain purple. Gram-negative bacteria have thin peptidoglycan, and they have an outer membrane. Archaea do not have a cell wall made up of peptidoglycan. If it is present, it's made up of other things. Mycoplasma bacteria lack a cell wall completely, and they're important because they cause something called walking pneumonia. The structure and function of the capsule. So capsules are made up of sugars, polysaccharides are the same thing, and they play an important role helping bacteria to stick to each other and to surfaces. And they also protect bacteria from being eaten up. We call that phagocytosis. So that's why they're medically important, because they're harder to treat. Flagella are important in motility. They allow bacteria to be motile. We didn't discuss this word cilia at all because prokaryotes don't have cilia, but in the next part of this lecture, you're going to learn about eukaryotes and they have cilia. And then pili, some bacteria have pili. It helps them exchange genetic material. We call that conjugation. How do bacteria move? They move because they have flagella. Where is the DNA located in the cell? It's in a region called the nucleoid. Under what conditions do endospores form? Endospores form in harsh environmental conditions, like if there's high heat or high radiation, that's when they form. Here, this one, why are drugs that target cell wall synthesis useful? They're important because, for example, if someone has a gram-positive bacterial infection, gram-positive bacteria have thick peptidoglycan. So if you give them an antibiotic like penicillin that targets the cell wall, it weakens the bacteria and the bacteria eventually die. The different morphologies of bacteria, the main ones are rod-shaped. Circular is called coccus, and spiral. And then why are Mycoplasma resistant to antibiotics that interfere with the cell wall? It's because they do not have a cell wall. Finally, I want to add one other thing that I hope you get out of this is that bacteria have a 7DS ribosome. And in the next lecture, we'll talk about eukaryotic cells.